In 1952, thalidomide was first synthesized by the chemical industry Basel Company. It was tested on mice and seemed to have no effects. The company was bought by a German pharmaceutical that was headed by Dr. Heinrich Muchter. He worked with Dr. Otto Ambros to perform testing and found that thalidomide had effects on rabbits and monkeys, and later found that thalidomide had sedative properties. They didn't test it anymore and deemed it safe for human consumption.
Pregnant mothers took thalidomide to help with morning sickness, believing that the placenta would keep children safe from whatever the mother ate or drank, including thalidomide. In fact, thalidomide didn't even need to be prescribed and could be taken whenever. It was learned that before 42 weeks gestation, the fetus could become severely affected by the thalidomide. After that amount of time, the fetus seemed to be unharmed. However, by the time this was discovered, many children had already suffered the consequences of thalidomide.
So, how did this happen? Well, thalidomide is what is known as an optical isomer. Isomers are molecules that have the same number and type of atoms, but are arranged differently. Optical isomers specifically are arranged almost identically, and in the case of thalidomide, there was only one difference. The two different isomers were distinguished only by a minute change in the orientation of a single nitrogen-hydrogen bond. The first isomer, R-thalidomide, was good at helping with morning sickness and sedation, while the other isomer, S-thalidomide, caused birth defects. The tiniest change between the nitrogen and hydrogen atoms on these molecules has created lasting impacts.
Many affected infants didn't survive long after childbirth, but those that did survive had serious birth defects, including loss or poor development of limbs, damage to internal organs, and bone hypoplasticity, among others. Some children were given braces and artificial limbs to help them in their daily lives. They faced greater challenges in school and learning, and it was difficult for them to adjust.
So, who discovered what was happening? Dr. Frances Kelsey was a scientist who finally found the connection between the mothers who took thalidomide and the affected children. As an officer of the FDA, she vetoed the approval of thalidomide in the U.S. market. Her choice was controversial until it was proven that the drug had caused the suspected damage. Thalidomide was finally taken off the European market in 1961, but by the time it was removed, it had spread to 47 countries and had affected over 10,000 children. Dr. Kelsey has since been lauded for her work in preventing further thalidomide cases. Dr. Kelsey also changed how the FDA approves drugs and made them require more testing to be done before they are made available to the public.
So, what are the effects today? We are still learning more about the properties of thalidomide. In the 1990s, it was found that thalidomide inhibited angiogenesis, or blood vessel formation. More recently, it has been tested and manipulated so it can be used in various cancer treatments and to prevent leprosy. But overall, the number of thalidomide survivors is less than 3,000 today, and although they have tried to get compensation, any help from the government is pithy. Despite this, survivors are still doing their best, and the FDA continues to test drugs more carefully before making them available to the public, thanks to the help of Dr. Frances Kelsey.